I welcome you all to another class on this in this school knowledge without the price. And in our previous class, we are talking about enzymes, and I explained explicitly what enzymes entails and its role in the body. And we looked at some of the functions of enzymes, relating it to health and what happens during disease conditions and how enzymes play a vital role in the detection of disease or in the detection of organ failure or progression of organ failure, and how enzymes play a key role in monitoring treatment. So if you have not attended that class, the link is down there on your screen. Make sure you go back and attend it. And for those students who follow suit to the end, there was a package at the end of the lecture. So please make sure you send in your responses to that email, and you'll be attended to as soon as possible. And if you have not subscribed to this page, I don't know what you are waiting for, because trust me, you are losing so much by not subscribing to this page. So make sure you follow us on YouTube, TikTok, Facebook, and Instagram to keep up with every knowledge that we'll be releasing on our platforms. So today we'll be looking at another interesting topic: carbohydrates. Carbohydrates is the topic for today. And talking about carbohydrates is not something that is so strange or something that we haven't heard about, because when we talk about carbohydrates, the first thing we have to take note of is that that is the fuel which the body uses. If we liken the body to a vehicle, then carbohydrates is the fuel needed to power up the engine. And carbohydrates is the main energy source of the body. And we'll talk about carbohydrates. We're talking about a particular nutrient that the body cannot do without. The body cannot produce it, so the body needs it so much in every of its activities. Now, one thing we have to note of is that, first of all, carbohydrates is made up of carbon, hydrogen, and oxygen. Let's go back to chemistry. While we're studying carbon, while we're studying the elements of the periodic table, they made mention of the fact that carbon is one of the elements that the body cannot do without. And the macronutrients, that is carbohydrates, proteins, and lipids, all of them have carbohydrates included in their structure. So it tells you that all of them have carbon, sorry. So it tells you that carbon has a vital role to play in the body. And we'll look at carbohydrates also. We'll talk about it as the major energy source of the body. And now carbohydrates are made up of simple and complex carbohydrates. Now we'll talk about the simple carbohydrates, we're talking about the monosaccharides. We're talking about the complex carbohydrates, we're talking about polysaccharides. Now I'll explain that in the lecture, so don't get confused. I encourage you to grab your pen, grab your notebook. It's going to get serious now. So as I mentioned earlier, carbohydrates, they're one of the major energy sources of the body. Now, when they are so concentrated in nature, when it is excess, you notice that it is being converted to fat. Reason why some people, when they consume so much carbohydrates, and don't forget, when we talk about carbohydrates, I'm not talking about sugars. Sugars is just an aspect of carbohydrates, but sugars is not a summary of carbohydrates. And now we'll talk about sugars. I'm not talking about glucose, because there's a misconception. Because when we talk about sugars, everybody tends to think glucose. Now let's snap out of carbohydrates. Let's talk a little bit about diabetes. Now we'll talk about increased sugar levels. We're not talking about glucose, because the misconception out there is that glucose is what causes diabetes, but that is not so true. Because when we talk about sugars, we are looking at monosaccharides, we are looking at disaccharides. All of them are sugars, not just glucose. So in this lecture, we're talking explicitly about carbohydrates and explaining what sugars actually entails and what they mean. And now, one thing about carbohydrates, or one major function of carbohydrates, is that at the level of the cell membrane, what they do is that they mix with proteins and lipids, forming glycoproteins and glycolipids, which aid in the transport of substances into and out of the cell, and also maintain membrane integrity and membrane fluidity. I explained all this in our previous lecture when I was talking about the introduction to biochemistry and the cell membrane. So if you want to attend that lecture, the link is below on your screen. You can just click the link and go back and attend the class, and make sure you jot certain things that is going to help you in this particular field. So that is what you have to take note of about carbohydrates. So in summary, the first thing is that carbohydrates are made up of carbon, hydrogen, and oxygen. The next thing, that the main energy source of the body. Excess carbohydrates are converted to fats to be stored up in the body. And now the fats are being stored up at the level of the belly, the groins. The reason why you notice that some certain parts of your body gets protruded with increased consumption of carbohydrates. As I explained earlier, Glucose is not what causes diabetes, or glucose is not the only sugar. Glucose can, but it's not the only cause of diabetes. I will talk about sugars. We are not talking about glucose only. Please take note. We'll talk about sugars. We'll talk about fructose, galactose, sucrose, and the like. We'll explain all of that in the course of the lecture. All right, so the next thing we're talking about is the nomenclature of carbohydrates. Now, I made mention when I'm talking about simple and complex carbohydrates, I made mention of examples like monosaccharides, disaccharides. Now, we'll talk about a monosaccharide. We are talking about a carbohydrate molecule with only one sugar ring. That's a simple structure of carbohydrates made up of just one sugar ring is called the monosaccharides, which can be hydrolyzed into smaller units. Now, fusion of two monosaccharides forms the disaccharides. Don't take notes. Monosaccharides is just a carbohydrate structure made up of one sugar ring. When it's made up of two monosaccharides, when there are two monosaccharides, it's called a disaccharides. When the monosaccharides are groups in pairs of threes, it's called a trisaccharides. When it's groups in pairs of fours and fives, we have a tetrasaccharides and the pentasaccharides, which gives rise to the oligosaccharides. Sorry. A combination of 10 monosaccharides, 10 or more monosaccharides gives rise to a polysaccharides. Now, if the combination of monosaccharides is made up of just one monosaccharide, we'll look at the different classes of the different molecules in monosaccharides class in the course of the lesson. So, but if the groups of 10 or more monosaccharides are made up of just a single monosaccharide, it is called a homopolysaccharide. But if it's made up of one or more class of monosaccharides, it's called a heteropolysaccharide. So that's what we have to take note of in when we talk about the nomenclature of carbohydrates. Now, the names of the sugars are groups, or the names of sugars are grouped dependent on the number of carbon atoms found in the sugar. For example, you have a triose sugar, a hexose sugar, a tetrose sugar, a pentose sugars. Now, all those sugars we face. Now, the difference between the sugars is just the number of carbon atoms that they portray. Because a triose sugar have 3 carbon atoms, a tetrose sugars have 4 carbon atoms, a pentose sugars have 5 carbon atoms. Now, before I continue, if you need the soft copy notes on this lecture or on any of our lectures, don't forget to drop a link or send us a mail and we'll send the soft copy notes through to you. Or you can click on our website and you go there, you have the soft copy notes there. Alright, so that is that about the nomenclature of carbohydrates. We have explained what monosaccharides, disaccharides, tetrasaccharides, pentasaccharides, and polysaccharides, all what they entail. So the next thing we're talking about are stereoisomers. So we'll talk about stereoisomers. We're talking about two molecules which have the same molecular formula but differ in the structural or in the spatial configuration of their atoms. So they have the same molecular formula, but they differ in the structural or in the spatial configuration of their atoms. Now, one thing about stereoisomers is that first of all, they have chiral carbon atom and they are optically active. Now, we'll talk about the chiral carbon atoms. We're talking about the carbon atom which is surrounded by 4 different functional groups. And we'll talk about optical activity. We'll be talking about it in our next class, in our next lesson, sorry, which brings us to the next lesson, optical activity. So we'll talk about optical activity of a particular atom. We are looking at the ability of that compound to rotate plane polarized light either to the left or to the right. Now, when the molecule rotates plane polarized light to the right, it is called a dextro rotatory molecule. When it rotates plane polarized light to the left, it is called a level rotatory molecule. And don't forget, if you have any question, make sure you either send to our email or you drop it as a comment. But I would recommend that you send it as an email so that you'll be attended to as soon as possible. And it's going to give us that clue that you are one of our serious students. And our molecules which are both level and dextrorotatory are called enantiomers. Now, there's a question I'll ask and I'll explain later, but I want you to drop the answer before you continue. Have you ever wondered why glucose is always written as dextrose? For those of us who are on internship already, why do we always see glucose as D-glucose or dextroglucose? Can you explain? Drop that, but I'll explain it in the course of the lesson. Now, let's look at the different types of carbohydrates in details. The first thing we'll be talking about are monosaccharides. We look at simple carbohydrates, talk about monosaccharides. Now, we'll talk about monosaccharides. There are four sugar molecules or sugar compounds that make up the monosaccharides. You have glucose, you have galactose, you have fructose, and you have mannose. Now, we will talk about glucose. This is the predominant sugar in the human body. Glucose is the predominant sugar in the human body. So whatever sugar you take in, it's being converted into glucose. And glucose, and the form of glucose which is being accepted in the body is the dextrose glucose. Don't forget, when I was talking about dextrorotatory molecules, I said they have the ability to rotate plane polarized light to the right. So the glucose molecule that is being accepted in the body, or that can be metabolized in the body, is the dextroglucose. The level structure of glucose can't be accepted or can't be metabolized by the body, so it is not absorbed. That is why in the hospital, the form of glucose that they give you is the dextroglucose. And now people usually ask the question, when people consume milk, not let's say pure milk without any additive in it, no sugar, nothing. Now they usually ask the question, is it that it's glucose that is in milk, or is it that it's normal sugar, as they put it? Is that it's normal sugar that is in milk? Why is this milk too sweet? That is because the sugar that is found in milk is different. The sugar that is found in milk is different. It's not glucose. The sugar that is found in milk is galactose. And when galactose is taken into the body, it is being converted into glucose at the level of the liver. So galactose is another monosaccharide. And now people usually ask the question, sugarcane is too sweet. What type of sugar is found in sugarcane? Is it glucose? What about pineapple? Because there are certain questions that arises. People ask, okay, we have glucose, we have sugar. True. But what about sugarcane? Yeah, is it glucose that is in sugarcane? Sugarcane that is too sweet. Pineapple, is it glucose that is in those foods, or in those fruits rather? But on the contrary, it's not glucose. It's another sugar called fructose. Fructose is a sugar that is found in sugar cane, and when it's taken into the body, it is still converted to glucose at the level of the liver, which is used by the body depending on the need in which the body has at that period in time. And if there is no need, it's being converted to fat when it is stored up in the body. The next thing we're talking about is anomalism of sugars. Now we'll talk about anomalism of sugars. Before I proceed, let me ask a simple question. Now, when we boil foods like potatoes, sweet potatoes to be precise, cassava, we'll notice that there are some parts which are they form a paste. They are edible. They appear, they taste good, and there's some parts which they taste as if they are poorly cooked or undercooked. No matter how you boil them, you always notice that that part is poorly cooked. Do we know why? Take your pen and paper and listen to me carefully. Now let's talk about anomalism of sugars. Now, anomalism of sugars is the ability of sugars or compounds to form a cyclic ring structure when placed in conditions which are not favorable, and it makes it stable. So we're talking about anomalism of sugars, the ability of sugars to form a cyclic ring structure rather than maintaining their long structures which can easily be broken. And now these cyclic ring structures give rise to alpha and beta sugars, like this way of an alpha sugars or beta sugars. Now we'll talk about alpha and beta sugars. Now after the formation of that cyclic ring structure, there is the movement of the hydroxide group either upward or downwards. Now once the hydroxide group moves upward, it is called an alpha sugar. Once it moves downward, it is called a beta sugar. Don't forget, I've still not forgotten the equation. Why is it that those carbohydrates with cassava, sweet potatoes, some of the parts appear edible, some doesn't appear edible, or some of the parts appear cooked, well cooked, and other parts appear undercooked? Why does that happen? I'll still explain it in the lesson. So jot that question when I explain it, and when I'm talking about the answer with, I will draw your attention back to the question so that you can take note of that. Now, the next type of sugars we'll talk about is amino sugars. Now, an amino sugar, just talk of a sugar which has an amino group attached to it. Now, the next thing we're talking about is oxidation of sugars. What happens when sugars are oxidized? Now, under mild conditions, the aldehyde group is being converted to the carboxylic group. So, glucose is being oxidized to gluconic acid, mannose to mannonic acid, and galactose to galactic acid. So that is what happens. Now, there are cases where, now the first case I talked about, that's under mild conditions. Now, in the case where the aldehyde group is being protected, the last carbon atom is converted to the carboxylic group, which leads to the formation of uronic acid. And glucose, in those conditions, glucose will be converted to glucuronic acid, galactose will be converted to galacturonic acid. That explains why in certain conditions you notice that glucose is being oxidized and being converted. Now, in the cases where you notice that instead of glucose you have gluconic acid, that's to tell that the aldehyde group has been converted to the carboxylic group. And in the case where you're having the glucuronic acid, that's to tell you that the aldehyde group was protected. Meanwhile, the last carbon atom was being oxidized to form the carboxylic group. Now, under strong oxidation condition, the first and the last carbon atoms are being converted to carboxylic groups, which leads to the formation of dicarboxylic acid, wherein glucose will be converted to glucosaccharic acid. So the different structures of glucose explains or gives reason for the condition or the names of glucose, the names which glucose possess, or the different structural formulas of glucose explain the conditions which they are under, either strong oxidation conditions or reduction conditions. So that is that about monosaccharides. The next thing we're talking about are disaccharides. And I don't forget, I explained that disaccharides are fusion of two monosaccharides molecule. Now, the first disaccharide we're talking about is sucrose. Now, sucrose is another sugar which is found in sugarcane aside fructose. Now, and sucrose is made up of glucose plus fructose. So sucrose is a combination of two monosaccharides, glucose plus fructose. And glucose or sucrose also, it is not a reducing sugar, means that it does not give up electrons to become oxidized. That's what you have to take note of about sucrose. The next disaccharide molecule we're talking about is lactose. Now, lactose is another sugar which is found in milk, and lactose is a combination of glucose and galactose, and lactose is a reducing sugar, which I explained when I was talking about sucrose. So it's a reducing sugar, it's a combination of glucose plus galactose, and it is found in milk. The next type of sugar we looked at, or the last type of disaccharide we talk about, is maltose. Now, maltose is the combination or the fusion of two glucose molecules. And don't forget, the fusion of two glucose molecules, it always leads, or the fusion of two monosaccharides always leads to the production of water at the end of the reaction. So maltose one is a reducing sugar, and it has an alpha or beta. It has both an alpha and beta nature depending on the configuration. And I explained what alpha and beta nature means when I'm talking about anomalism of sugars. So that is that about disaccharides. The next thing we are going to look at are polysaccharides. Now, polysaccharides, there are two types. I mentioned polysaccharides in the beginning of the lecture. I mentioned homopolysaccharides or homoglycans, and heteropolysaccharides or heteroglycans. Now, homopolysaccharides or homoglycans are polysaccharides units that are made up of just a single monosaccharide unit. For example, I said earlier that polysaccharides combines are made up of 10 or more monosaccharides units. Now, if these monosaccharides units are made up of just glucose, or just fructose, or just galactose, or just mannose, it is called a homopolysaccharides. But now, if this group of monosaccharides are made up of maybe glucose, fructose, glucose, mannose, glucose, galactose, it is called a heteropolysaccharide. That's what you have to take note of. And now I'll talk about homopolysaccharides or homoglycans. We look at starch, we look at glycogen, we look at cellulose. And we'll talk about heteroglycans, we look at higher uronic acid, and we'll talk about chondroitin sulfate. So let's talk about, let's talk about the homopolysaccharides. The first homopolysaccharide we're talking about is starch. And starch is the reserve polysaccharide, or the reserve storage for glucose in plants. So that's the reserve carbohydrate storage in plants, and it's made up of two types. It's composed of two classes. You have amylose and amylopectin. Now, when starch is placed in water, the solubilized part is amylose, and the part which is not soluble, or which does not get dissolved, it is called amylopectin. Okay, now let's go back to the question I asked. Why is it that there are certain food, let's take an example, cassava, when you boil it, you notice some parts are edible and some parts are not. Now, don't forget cassava is starch, and made up of two parts, amylose and amylopectin. Now let's go back to anomalism of sugars. I explained that with anomalism of sugars, when it's placed in unfavorable conditions, especially water, it forms cyclic ring, which helps it, which helps it not to dissolve or not to form a soluble paste. And don't forget, I made mention of the fact that starch is amylose and amylopectin, and amylose forms a soluble paste while amylopectin doesn't form any soluble paste. That's to tell you that amylose is the part that gets solubilized, while amylopectin is the part which forms the cyclic ring, or is the part which undergoes anomerism. So when it's placed in water, amylose doesn't undergo an anomerism, while amylopectin undergoes anomerism. Reason why it doesn't form any soluble paste, and reason why amylose forms a soluble paste. So next time when you boil cassava, I notice that a part appears undercooked or polycooked. Now that part composed mainly of amylopectin, and the part which is cooked properly contains or composed mainly of amylose. And examples of starch, as I explained earlier, we have potatoes, especially sweet potatoes, we have cassava, we have plantains. And majority, amylopectin makes up 70 to 80% of starch, while amylose makes up 20 to 30% of starch. Now, there are cases, now there are some, when we say 70 to 80%, but there are some foods, starchy food that you will get, and majority of them will be amylose, and there are some that majority of it will be amylopectin. Now, amylose is made up of straight chains, while amylopectin is made up of branch chains. I know that when it's made up of branch chains, the probability for those bonds to break is not so feasible. And amylose is made up of is linked by an alpha 1,4 glycosidic bond, while amylopectin is linked by an alpha 1,4 and alpha 1,6 glycosidic bond. So you see that the branching in amylopectin makes it a little bit complex for it to be soluble. Reason why you notice that when you boil those foods, as I explained earlier, it is unedible or appears undercooked. So if you have gotten the answer, if you have understood the answer, please comment understood and type the question. Just comment understood beside the question in the comment section. Thank you very much. So the next type of homoglycan I'm talking about is glycogen, and glycogen is the reserve carbohydrate in animals. And animals are talking about humans, and glycogen also is stored up in the liver. Excess carbohydrate is stored up as glycogen. That's what you have to take note of. And glycogen is composed of alpha dextrose glucose. Take note. Glycogen is composed of alpha dextrose glucose. Reason why, which explains why when glucose is needed in the body, it's the dextrose form that is being given, and it is made up of it's linked by an alpha 1,4 and alpha 1,6 glycosidic bond, and it is highly branched, which accounts for its highly resistant nature to adverse conditions, maybe water, maybe heat, and all the like. Now, the next polysaccharide I'm talking about is cellulose. And before we dive into this lesson, please, I want you to write down this question because we'll be answering it in the course of this lesson. Now, have you ever wondered why we consume any vegetable, irrespective of the vegetable, your physics is green, but when you consume starchy food, other foods, let's say for example, let's say today you consume, let's say huckleberry or arrow, you notice that your physics will represent that same color of the vegetable. Sometimes you even find leaves, patches of leaves in your physics. But when you consume other food, let's say cassava, let's say rice, your physics is different. Have you ever wondered why? Okay, grab a pen, we'll be talking about it in a few minutes. Now, one thing I take note of about cellulose is that it's the supporting tissues which is found in plant, and it's also a connective tissues. And cellulose is made up of beta dextroglucose. Take note. First thing is made up of beta dextrose glucose, and it's linked by a beta 1,4 and beta 1,6 glycosidic bond. And the main reason why we can't digest cellulose is because we lack the enzyme cellulose. Now, the absence of this enzyme makes the body unable to metabolize and absorb cellulose. Reason why when we consume vegetables, the vegetable goes through our physics the same way it was taken into our mouth. So I believe you have understood the question, or you have understood the answer to that question. We lack the enzyme cellulose, as opposed to other ruminants like sheep, goats, which have that enzyme. Reason why they can consume those vegetables. For example, those who have goats and or other ruminants, you notice that when they consume those green leafy vegetables, their physics is not the same as the vegetables, as opposed to us, which our physics possess that same color as vegetables. Now we'll go into the next class of polysaccharides. Those are which we're talking about heteroglycans, and under heteroglycans we'll be talking about mucopolysaccharides. Now, when we talk about mucopolysaccharides, we're talking about heterosaccharides, which contains both uronic and amino sugars. And don't forget, when I talk about uronic sugars, I'm talking about the case where do you looking at the oxidation of sugar, where it's the last carbon atom which is being oxidized to the carboxylic group. Reason being that the aldehyde group was protected, and now when it happens, it gives rise to uronic sugars. And amino sugars are carbohydrate molecules which have the amino groups. So under uronic sugar, we're looking at higher uronic acid, and we look at heparin. Now, talk about higher uronic acid. Now, first of all, this is a connective tissue that is found in tendons. It's found in connective tissue. Sorry, higher uronic acid is found in connective tissues, tendons, and synovial fluid. And we know that all these aids in flexibility of joints and also help to absorb friction. Reason why you notice that your bones aren't sliding against each other, and when you exert certain pressure on certain surfaces like this, you notice that there is no dislocation anything of that sort because of the presence of this compound, and it also serves as a lubricant in cavity joints. And the next class of mucopolysaccharides that we're talking about is heparin. Now, heparin is one molecule or is one compound that prevents coagulation in blood and helps to prevent thrombosis. And that is what happens. And the question of heparin, when you look at patients with SS, it's very low. Reason why you notice that there's high probability for them to always experience thrombosis when it comes to their blood. So that is that about heparin. It's an anticoagulant. The next thing we're talking about are glycoproteins and mucoproteins. But don't forget, if you want to know more, here is just a summary because I took time to explain it in details when I was talking about the cell membrane. So here is just a little summary of what glycoproteins and mucoproteins entails. But if you want to know more about it, the link is down there on your screen. You can click it and go watch our lesson on Introduction to Biochemistry, where I explain explicitly what all of this entails. So let's look at glycoproteins and mucoproteins. So glycoproteins, first of all, glucose molecule to proteins, and I explain what mucoproteins are all about. So a protoglycan, let's talk about a protoglycan. Now, a protoglycan is carbohydrate molecules which have been added to a polypeptide chain. Now, when carbohydrate molecules have been added to a polypeptide chain, it is called a protoglycan. Now, if the carbohydrate content of this protoglycan is less than 10%, it's called a glycoprotein. If the carbohydrate molecule in the protoglycan is more than 10%, it is called a mucoprotein, and they are found in all tissues and membranes, especially the cell membrane, and play a vital role when it comes to transport of molecules into and out of the cell. And now the carbohydrate molecules that covers the surface of the cell membrane are called glycocalyx. Now, and the synthesis of complex polysaccharides in bacteria is blocked by penicillin. So that's why you have to take note of glycoproteins and mucoproteins all arise from the protoglycan, which the protoglycan entails carbohydrate molecules which have been added into the polypeptide chain. That said, we have come to the end of the lecture on carbohydrates, and I encourage you to drop in your comments. And I want to know how the lecture has helped you. I want to know what you have learned from this lecture. And as a package from Knowledge Without the Price, there is a quiz that we are posting at the end of this lecture, which I would like every student to answer and send in their responses to our email, which will attend to, which those responses were attended to as soon as possible and forwarded back to you through your email. Don't forget to visit our other platforms on Facebook, TikTok, Instagram, and also like, share, and comment. And most especially, refer someone to this page, because you can't be enjoying all this alone. Trust me, someone else needs to learn and someone else needs to be updated on this knowledge. Thank you so much.